Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It is our great pleasure today to have Dr. Santiago Aja-Fernández to give us BrainMap talk. Dr. Santiago Aja-Fernández received the Engineer of the Communication and the PhD degree from the University de Valladolid, Spain in 1999 and 2003, respectively. He's a full professor with the ETSI Communication, the University of Valladolid, where he's also the Laboratory of Image Processing. His research interests include medical imaging analysis and processing with special interest to MRI, diffusion MRI, biomarkers, and clinical applications. He was awarded with the Fulbright Scholarship for 1 year stay as a research fellow with the Laboratory of Mathematics in Imaging at Brigham's and Women's Hospital, Harvard Medical School, Boston, from 2006 to 7. In 2019, he spent 6 months as a visiting scholar in CUBRIC in Cardiff, UK. I would like to remind our audience to address their questions by raising their hands or by using Q&A box at the end of the talk. Thank you for coming today, Dr. Santiago Aja-Fernández, and the stage is yours.
Well, hello everybody, the ones here and the ones online. If along the talk you feel like you want to ask about any slide, or what do you want me, to clarify anything, you can do it there, or you can do it at the end of the talk. It depends on how you feel about it. So I'm going to talk to you about artificial intelligence, medical imaging. And it was not a workshop, it was a challenge we did a couple of years ago, trying to, the idea was trying to understand what was happening when we were using artificial intelligence in medical imaging. We know that pictures are very fancy and very realistic, but can we use that on a real clinical study? So we tried to answer that, asking many groups to do exactly the same. And here are some of the results, and I think very interesting implication of the use of artificial intelligence. So first of all, why are we interested in this? So if you search artificial intelligence or AI-generated images online, you can find lots of images. And the four ones are not, really not the real images, but if you check it, they seem quite realistic. They can be actual pictures you take with your phone or your camera. And these two here in the bottom, there are two people that don't exist. Just generated by artificial intelligence. So there's a page, a web page, in which they generate lots and lots of faces of people, non-existent people. So in the end, if you check these pictures, they are quite realistic. They can be real people. There, say, okay, or you can be there. But the thing is, are they real? They are not real. They've been generated. They are not real. In reality, they never occurred. But the pictures can be real. So let's translate this into medical imaging and keep this in mind, the real versus realistic. So we have been talking a bit about deep learning, medical imaging, clinical studies, and specifically diffusion MRI. This study is focused on diffusion MRI because it's the modality we are working on, but I think some of the conclusions can be extrapolated to other kinds of medical imaging, I think. I assume most of you have gone recently, and I say recently, I say the last 5 years, to conferences about medical imaging or magnetic resonance imaging or whatever. And if you go there, you can see that every year there's more and more papers about deep learning or artificial intelligence, lots of them. Maybe it's changing a bit now. I do remember Mikai, for instance, like 5 years ago, 90% maybe of the papers were about deep learning applications. Actually, I think they have this policy now in which they say no more than 50% can be about this, just to have I mean, space for all other kind of things, because if not, I mean, they were a bit repetitive. But if you go, you can see this. Actually, pictures generated by, when I say pictures, I say in the brain or MRI or a slide or whatever, generated with artificial intelligence, they look very good, very realistic. And usually to test the quality of these images, they use quality measures that usually are also employed in general images, like the similarity index or some kind of error, minimum square error, or whatever. So they have very good metrics in this index, like saying, okay, this is very good picture. It's 94% similar to the original image. But can we apply these techniques to real medical studies, or is there any risk of losing relevant information? Are we losing information that is key for our study? And that's what we were trying to understand here. So the question we posed for this challenge and for our study was, are we losing relevant quantitative clinical information when generating high quality images with artificial intelligence techniques? In lay terms, if you use artificial intelligence, deep learning, what we get usually is very nice, but are we losing some information that we cannot see with the eye? Is there information there missing? So that's where we wanted to test or to validate. So the main questions, there are three main questions we wanted to answer. First of all, are deep learning techniques, when I say deep learning, can be even broader. We can, one of the teams use machine learning, but these artificial intelligence techniques are able to improve the quality of the fusion MRI data to be used in clinical studies. I mean, just not for fancy looking, but just for clinical studies. Are the subtle details and differences between groups kept or lost when using deep learning? And finally, if they are good for these clinical studies, which of the method is best? What kind of approach to improve your data is best to be used in clinical studies? So we proposed this challenge in MICAI 2022. I'm going to talk a bit about it later, but I mean, there were 36 teams downloaded training data. Finally, 15 teams submitted the final results. One of the teams submitted 2 different methods. So I think what was quite a success. And in addition, I mean, we had very different points of view of how to deal with this. So I think it was good. And in the end, more than 40 researchers participated in the final paper and the final results. If you're interested, I mean, the paper was published this year, so you can look for it, and everything I'm going to talk is there. But today I want to talk a bit more about the insight. So we decide to use first diffusion MRI, because it's the thing we are working on and we are familiar, and we had a specific problem and we wanted to solve it. And also we use migraine. I can say many things about migraine, but basically we use migraine because we had a very good database of migraine. So we can use it. We have lots of cases, and we have probably there are other diseases that can also be used, but I mean, this is the one we had. The good thing is we had a very good database with many cases, and we use in a specific, when we acquire data, we always, this is something we started to do in the past because we needed for a specific problem, and we kept doing it like the last 10 years. So we have, I don't know if you're familiar with diffusion MRI, I assume most of you are, but we have this scheme of gradient directions, of 61 gradient directions that are optimized just to be subsampled to 40, to 21, or to 6. So just acquiring 61, we can decide to use just 6, and they are properly spaced on the Q space, and 40 and 21. So we can do some downsampling and to use this for benchmarks and things. We decide also to use chronic migraine versus episodic migraine, which is the clinical study we are doing, because we detected that there are differences, but that very subtle differences. If you change the number of gradients, if you change the number of samples, if you change the number of subjects, you're going to lose lots of information. So I think it was kind of very nice data set to work because of this. And I mean, I said last line, if you change the number of subjects of the number of gradients, all the differences go down. This is one experiment we did in the past in which for a specific method of measure, but we decide to change the number of directions when we do the comparisons or the number of subjects per group. And you can see the number, this number here is the number of regions with significant differences after some kind of statistical test. And if you reduce the number of gradient directions, you reduce the number of differences. And if you reduce the number of subjects per group, you also reduce the number of differences. So if you are not careful with this study, you can lose all the differences there because they are very, very subtle. You need lots of subjects and very strong information there. So that's why we decide to go with this. This is an example of the kind of sampling we do of the Q space. I mean, you can, but I mean, doesn't matter here. So the experiment was something like this. I mean, if you have some subjects with chronic migraine and some subjects with episodic migraine, they go into the scanner, and you have in the top, you can see 61 gradient directions acquired, and you can simulate 21 because we have this special scheme. And the first row, you do the statistical analysis, and you have some differences in FA and D and so on. And you can also do the differences here in the bottom line. I mean, with 21 gradient direction, you do exactly the same. Actually, all this registration and so on, we do it with the 61 gradient and extrapolate to 21 just to do it exactly the same. Just we need just the variable here is just the 21, I mean, the number of gradient directions. And I have this table here, I mean, in which basically you just have to look to the bottom line, in which you can see that the number of true positives, I mean, the number of points in which you detect differences when you do TBSS is like 40% of the points with 21 gradients. So you're losing around 60% of the points you have when you decrease the number of gradient directions. So this is going to be our problem. I mean, if you reduce the number of gradients, you lose lots of information when you do a clinical study, right? And you also have some small number of false positives, but very, very small there. So the question, I think, is here is, you can wait a second, with these numbers. Can we improve those numbers? Can we go closer to the 17, for instance, in AD? Can we go closer to, I mean, from 7,000 to 17,000? Can we go up there by using artificial intelligence? Can we improve the data and try to get something better with a fewer number of gradients? And that's the challenge we propose. So we are going to ask the teams to give us just FA, MD, and AD. And this data is from a real study already published. We know where the differences are and so on. And we are going to ask them to improve the data or upgrade the data diffusion data for 21 gradients to 61 gradients. They can interpolate the gradients or they can just generate the FA similar to FA of 61 gradients. I mean, the method doesn't matter. Anyone can do whatever they want, but we ask them just to give us this FA, MD, and AD. Since we are losing 60% of the difference, the question is there any deep learning method that is going to be able to recover those differences, or at least to improve a bit that? So we have this data. So we gave them 60 healthy subjects with the 61 gradients and 21 gradients. Each subject, I think there are like 92 slides. So, I mean, there's lots of data there for training. And in addition, we gave them 50 migraine, 50 plus 50, I think, 50 episodic migraine and 50 chronic migraine patients with 21 gradients. And this test data is totally blind. I mean, they don't know which ones are episodic migraine and which one are chronic migraine. Actually, they are shuffled there and they have no idea. And we just gave them 21 gradient direction. So the idea some people say, okay, but you should also train with chronic migraine or episodic migraine to improve your method. Yeah, but our idea is there are now commercial scanners that are going to give you blind artificial intelligence methods to improve the quality of your data. And it's not trained with, I don't know, schizophrenia patients, migraine patients. No, they are also trained with healthy subjects. So that this is what we want to do here. Train with healthy subjects, trying to improve your data, and let's see what's happening when you apply that to a specific disease like the migraine here. So the method should be something like this, that you have the patients, you can train with 61 gradient and 21 gradient of healthy controls and generate the final images. We are going to do the clinical study, not them. They gave us the data. We do exactly the same study or the statistical study for everyone, and we obtain results. And we have some metrics to compare the quality of the results. So sorry if I'm a bit long here explaining all these details. I think the interesting part of the results, but I think I wanted this to be clear. So as I said you before, we are going to give these data sets, the trainings, the subjects, the validations, which is blind and so on. And the participants, there were lots of groups, 36 groups download the data. I said before 15 submit results. Actually, it was 15, but one of them was so bad results that we, exclude that group because it was totally, I don't know what they did, but it was totally nonsense when they submit. But some of these groups, I think there's one here from Martinos, there were another one from MD8, I think. I don't know if any of you were here. Anyway, so many different universities. Some of them couldn't submit it on time, but initially 36 groups were interested in doing this. You don't need to read everything, but this is just to have an idea. This is in the paper, to have an idea that each team proposed totally different approach. Some of them, the first one was just machine learning, not even deep learning, actually with very, very good results. We have a convolutional neural networks, other methods. Most of them have the DWIs as input and DWIs at output, and after that they generate the FA or whatever. But some of them, they just generate the FA with the network. Many different approaches, many ways of training. Some of them use just have to train and have to test, other uses. There are many different combinations. And I want to recall this because results are going to be very funny. Because, I don't know if funny is the word, but have in mind that there are many different combinations, and just a couple of methods were a bit similar, but most of them were very, very different. Actually, some of the groups use methods to the noise. Networks prepared to the noise. Actually, when our data was already the noise, but they use the noise method. Others use some network prepared for video, just to interpolate frames in video, and they use that. Very different approaches, some of them very creative. And let's see the results. I'm going to show you, when you see a table with lots of number, don't be afraid, don't worry about it. Just going to just to have a bit of perspective, but I'm not going to explain all the numbers there. First of all, if we go to visual results, as I said before, most of them seems realistic. The FA seems FA, the AD seems AD, and the MD seems an MD. Actually, the first and the second are without, the 61 gradient generated and the 21 gradient generated, which actually are from the real data, and the other ones are generated by this method. There's one method in Team 9 that I don't know exactly what they did, these guys, because the method was good, but somehow they make, I don't know why, but they did some kind of change of the level, and all the results are like, half of the values they should have. It doesn't matter when you do a clinical study and you have scaled everything. So, it didn't affect a lot, but visually, if you see Team 9 is the only one that is a bit different because it's a bit darker because of that, because I don't know why. Once more, I think they have some problem with the scale there. But the other ones, you can see they're, quite realistic if you make a zoom and say, okay, most of them are quite similar, and I cannot see any differences. And this is one of the keys of this work, of this experiment, is if you take a look like this, you say, okay, this is realistic. That seems like an FA. Okay, of course, this and this. So are we losing their information? I mean, if I do a clinical study with this, it's going to work? Visually, I cannot say anything. So if you go to the numbers using the PIC-SNR, and I don't know if you're familiar with the SSIM, which is the structural similarity index for images, which is a standard image. When you compare two images, you're doing the noising, for instance, or you're doing reconstruction or whatever. You want to compare two images. This is an index that tells you how close they are. 91 would be they are exactly the same. If you go down, they are quite similar. Depends on that number. So it's usually very common index for in artificial intelligence and in reconstruction. And if you can, as a reference, I did the comparison between the 61 and the 21. So all the numbers in red are worse than doing nothing, and all the numbers in black are equal or better. But if you check, we are starting in a very, very high value. I mean, 90%, 91, 94 for SSIM. So most of the methods are already there, some of them a bit worse. So using these numbers is not telling anything about the goodness of the method. I mean, if you check that, see, oh, there are some methods that they are even worse. Team 9, if you check, which is the one that changed the level, it's, I mean, very, very bad, because, I mean, the level is totally different. But if you check, the other ones are more or less the same values. So the first thing that we should take into account is that these classical structural methods or structural metrics are not going to tell you anything about the goodness of your method, or at least it's not very relevant. Most of them, they are going to be very similar. But once more, doing nothing is also very good. So what are we doing anything here? So I think I have this very, very long table here. I think we can skip a bit this. What we do, I'm going to tell you better this later, but just compare the numbers. What we are going to measure is more important what's are the results. So we try to see how many true positives can we find, and how many true false positives, sorry, can we find. So, and we compare that to the numbers with 21 gradient directions. So we use lots of different metrics for that. ACC, I mean, there are traditional numbers that, forget about the numbers, but these are traditional metrics of how good your metric are, how close to the original. And I have getting all these numbers and I put it just in one table, ordering by, I did like a special equation just to take everything into account. And so in the end here, they are ordered as how, with a number, if you see the second column, I compare to 21 gradients and I say, how much do I improve with this method versus doing nothing? If I do nothing, it's the 21 gradient direction, which is 0%, and I improve a 1.4%. In the first one, I improve a 31.8%, I mean, around 32%. So it seems that most of the method, I mean, if we take out the three last, most of the methods are improving a lot the results. So it seems that, okay, using artificial intelligence can be something good. I mean, I find new differences that they're lost when I reduce the number of gradients and so on. What is interesting here is just check the purple columns, I mean, true positives and false positives. I increase in the I finding in the first one, almost 77% of the original true positives, when by doing nothing, I get 40%. So it seems like a great increase. Okay, I'm doing something good using artificial intelligence. But if you go to the red number in the FP, false positive column, you can see that the more true positives I find, the more false positive I find. And if you check that, it's almost linear there. I mean, if I'm getting more and more and more and more true positives, I'm also getting more and more and more false positives. And I'm going to see that later, because a huge relation there and give us the idea that are we actually finding those differences, or are we generating random differences, and most of them are in the areas I want to, I look in. And that's a question we should ask when we are using this kind of data. In addition, also take a look to the red columns, the quality metrics. I mean, the green ones are improving and the red ones are doing it worse. And if you compare to these columns here, it seems that there's no correlation at all. I mean, the first two teams, they have very bad quality metrics. And the ones with very, very good quality metrics, some of them are in the bottom. I mean, check the team 4, which is the 3 by the bottom, is the one with the best values there. And I mean, and it's doing it worse than the original one. So it seems that then there is no correlation between this usually very used quality metrics used for images and what we really have when we are applying this to a medical or a clinical study. So we have to be very careful when we validate our deep learning method with this kind of metrics, which is very standard, and we did it in the past. And we, for instance, we have a student doing generating flare images from T1 and T2, and with a very, very good quality and very similar to the original images, but very similar using this kind of metrics. We didn't apply to a real study. Probably if we go to a real clinical study, the numbers can change. And what I'm going to show you now is some pictures that I think are going to illustrate this much better. I'm getting just a couple of metrics, the AD and MD. And in blue, you have all those points in which you find differences in 61 and in 21. I mean, those are the numbers found also only by 21. In green, those that are not in 21, but are found by the different methods. And in red are the false positives. If you see in all of them, the longer the green, I mean, the first one is the original one, is the reference, so there are no false positive. But the other ones, if you check, the longer the green line is, the longer the red line is. So it seems very clear that when you're finding, for example, check teammate, if you're finding more original points, more true positives, you're also finding lots of false positives. And in a clinical study, a false positive is something that, actually you don't really want. I mean, you can find differences in some areas in which you don't really have differences. And that's crucial when you are looking, for instance, in migraine, where in the brain is the migraine affecting, which are the areas we have to look, where are really the differences there. And if you do this one, we can say, okay, but it can be that there are really differences that cannot be found, but cannot be found by the original method, but deep learning is so good that they discover new differences. So let's say, okay, let's see if those differences are in the same areas for the different methods. And when we check, this is the number of teams that have one difference in 1 point. So what we are seeing here is that most of the points we find are just in one team. And not all the points are in all the teams. So what we are saying is that the false positive we are finding, they are randomly distributed across the teams. They are not all in the same place. They are totally random. And this is, I think, very, very interesting. When you make a plot of the number of false positives and the number of true positives you find with this method, and remember that each method is calculated with the same data, but different teams, different methods, different areas, of course. And the results are quite different for all of them. And if you plot this, you have almost a line here. For ND, it's not totally line, but here you have an R2 of 0.9, which is extremely high. I mean, if you do any kind of statistical analysis and you have this R for any kind of disease or whatever, it should be, you reanalyze the data because you think probably I've done something wrong. It cannot be this high. But for this number is very, 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 very high. And it means that if your number of true positives grow, your number of false positives also grow with it. So be careful, is what I can say here. Be careful. I cannot say that this is going to apply for all the methods, I mean, all the applications, but for these specific applications, I mean, for at least for me, it was some big red flags saying, be careful when you use deep learning or artificial intelligence methods, because the better your method is working, the more false positives you are generating here. Lots of them. And it's amazing that it's almost linear, and I say it again, its point is different methods and different teams and different even perspectives, very, very, very different ways of dealing with the data. Of course, the data is the same and the purpose is the same, but the methods are totally different, the rounding is totally different, the filtering is totally different. And in both methods, we have this very, very straight line. So I don't know if being a bit fast here, I mean, anyway, we can talk later more about it. So what are the conclusions of this thing? Most method, I mean, I have to say before this, that when we proposed the challenge, we were expecting that all the methods are going to perform worse than doing nothing, because we did some kind of fast experiments before that. We had some methods already implemented that can be applied to this. So we tried, and we have very, very, very bad results. So we say, okay, let's see if everything, I mean, all the teams are going to do this very badly. So our expectation was saying, okay, we are going to show that artificial intelligence is not working and all the methods are very, very bad. So it was a surprise to see that most of the method improve the quality of the final images, and most of the methods succeed in finding new true positives. And different methods perform differently. There are some methods that work very good and others not that good. Actually, the one with better results was one just using machine learning and doing some kind of interpolation of the Q space. This guy trained with, I think, with four volumes. Just instead of getting the 60, just got 4 of them just to test a bit the interpolation. But this was like a numerical interpolation. He didn't try to learn from the data, just try to see how to interpolate those points. And it was the one working, I mean, it was the simplest one, the fastest one, and the one with the better results. So in terms of more true positive and less or fewer, sorry, false positive. The second conclusion is that the global image quality metrics like the peak SNR or the minimum, the mean square error or the structural index are not adequate for evaluating the quality of this kind of data sets if we want to use them later for statistical analysis of a clinical study. If you want to have a very nice picture, probably they're going to be very nice metrics. If you want something to be very similar visually to other images, or something to be watched for by a doctor or by someone who is going to evaluate your grant, probably these kind of metrics are very good because you are going to have very, very nice images. But for clinical studies, at least in this case, and I think probably we can extrapolate it, I don't think they are the best, probably the best kind of metrics. And sometimes you use these metrics as quality for generating your images. So probably we should also have this into account when generating, using deep learning and generating and learning from the data. Once more, in this case, artificial intelligence-based processing of MRI data could significantly increase the number of false positives. And if you want to do clinical studies, this is very important. Once more, is the same. I think the debate here is, or the discussion is exactly the same we had like 10 years ago about filtering or not filtering the data. Are you losing when you filter your data, you denoise your data? Are you removing information? Are you adding new information? Because sometimes you find more differences, sometimes you lose difference, but are those differences really there, or they are just noise? or whatever. So, I mean, it's going back to the same debate once more. But I think we have to keep it in mind. Be saying, okay, if you want to apply these methods, you have to be very careful. And if you check your scanners, more and more the vendors they are including AI-based processing methods that increase the quality of your data. At least, I mean, Siemens has it, and I think Philips, we have a new computer to control our Philips scanner, and they introduce some new, very advanced, but artificial intelligence based in quality improvement and so on. Be careful with that, because those are not trained with specific diseases, but those are trained with usually with healthy controls and with different databases. So sometimes the side effect of using those methods is exactly what's happening here, that you remove part of the differences, but in addition, also you can generate new differences. And finally, I mean, it's the same, I just said number 4, general purpose methods based on artificial intelligence have to be taken with great care, because, I mean, can be a bit deceiving. I mean, can be careful with that. At least try to validate your methods in a clinical environment, or at least try to validate your methods with the purpose of the task you want to do. It's not the same if you want to have a very nice illustration for a paper or for a book, that if you want to use it in a clinical study or multi-center study or whatever. Finally, I mean, of course, I was not the one doing all the work. This is the team from my university that was behind all the analysis and preparation of the data, taking into account that we have to do all the statistical analysis of the data of each of the teams were done by my team, and because we wanted to do everything exactly the same. So just we do all. I think it was a lot of work for these guys. So we want them also to be here. And I think that's it. I mean, I've been a bit short maybe, but I don't know if you have any questions or comments or whatever.